There are several different organelles that make up the cell. One of these, and the most important, is the nucleus. The nucleus can be found in all eukaryotic cells and can sometimes even be seen in multiples like in fungal cells. The nucleus is the largest cell organelle at 6 micrometers in diameter. It consists of a double membrane, also called the envelope. The nuclear envelope contains pores that control the exchange of substances between the cytoplasm and the nucleus. Hormones like estrogen and thyroid T3 have receptor molecules inside the nucleus and needs to be able to enter the nucleus. Other molecules that can enter the nucleus include ATP, nucleotides, and proteins that are needed for ribosome synthesis. Ribosome synthesis occurs at the nucleolus. It uses information from its own DNA, and the ribosomes are then transported out the nucleus to enter the cytoplasm or the endoplasmic reticulum. The nucleus also contains DNA in the form of loosely coiled chromatin wrapped around histone proteins, except for during cell division, where the chromatin will be seen as condensed chromosomes. The DNA in the nucleus contains genes that can be transcribed into mRNA. mRNA will then be transported out the nucleus to ribosomes to be translated into proteins. The overall function of the nucleus is to store DNA, which is responsible for controlling the activities of the cell. Continuous with the outer membrane of the nucleus is the rough endoplasmic reticulum. It is named as such because of the rough texture caused by ribosomes that are embedded on it. It forms an extensive system of membranes that run through the cytoplasm with flattened sacs where chemical processes can take place separate from the cytoplasm. The rough endoplasmic reticulum is responsible for protein synthesis. The protein that is manufactured can enter the sacs and move through them. This can sometimes alter the proteins themselves. Small pieces of sacs containing protein can break off, forming vesicles. These vesicles can then combine together to form the Golgi complex. This all forms part of the secretory system. Let's have a look at an electron micrograph of the nucleus found in a pancreatic cell of a bat. We can clearly see the nuclear envelope with the nuclear pores. The nucleolus is darkly stained, and the loosely coiled chromatin can be seen as a granular appearance. We can also clearly see the endoplasmic reticulum surrounding the nucleus. This gives us a good picture of how large and widespread the endoplasmic reticulum is. Elsewhere in the cell, we also find the smooth endoplasmic reticulum. The only difference from the rough endoplasmic reticulum is that it does not contain any ribosomes, so it will not be able to produce proteins. Instead, it's responsible for synthesis of lipids and steroids, like cholesterol and reproductive hormones. Here is a photomicrograph showing a good comparison between the smooth and the rough endoplasmic reticulum. Here in the upper left corner, we can see severe branching of the smooth endoplasmic reticulum. It connects with the rough endoplasmic reticulum that can be easily identified by the ribosomes on its surface. Like previously mentioned, vesicles can bud off the endoplasmic reticulum and combine together to continuously form the Golgi body at one end. At the opposite end, the Golgi body will be broken down to form new vesicles. Together, the Golgi body that consists of flattened sacs and the vesicles are known as the Golgi apparatus or the Golgi complex. Now, multiples of these sorting and distribution centers can be present in a cell and are responsible for several different modifications of biological molecules. One of the most important modifications include the removal of methionine from a polypeptide chain to produce a functional protein. Now, the amino acid methionine is produced by the start codon AUG and does not contribute to the functionality of the protein. Other examples include the addition of sugar to protein molecules to produce glycoproteins and the formation of lysosomes. Lysosomes are 0.1 to 0.5 micrometer spherical sacs surrounded by a singular membrane with no internal structure. They contain hydrolytic enzymes and are responsible for the breakdown of unwanted structures like organelles and sometimes even whole cells. In leukocytes, they are used to digest bacteria, and in sperm, they digest a path to the ovum and is known as the acrosome. The Golgi apparatus is also important in plants, as it's responsible for converting sugars into cell wall components. Here we can see the Golgi complex, which consists of the Golgi body as well as its vesicles. It can be distinguished from the endoplasmic reticulum as it's much smaller. Next, we have a reconstructed 3D model of a mammalian cell showing the interaction between the endoplasmic reticulum and the Golgi apparatus. We enter the cell clearly seeing the enormous nucleus. Closely surrounding it is the endoplasmic reticulum, where different biological molecules are produced. Vesicles will bud off the endoplasmic reticulum and travel towards the Golgi apparatus, where it will fuse with its membrane, and here it can be modified and transported elsewhere in the cell, or it can even be exported to the outside of the cell. Also found in all cells are ribosomes. They can either be seen floating in the cytoplasm or embedded onto the rough endoplasmic reticulum. They are made up of ribosomal RNA as well as protein and consist of two subunits, with the larger subunit located on top of the smaller. The size of ribosomes, known as S units, refer to how fast they sediment in a centrifuge. 80S ribosomes are seen in the cytoplasm and endoplasmic reticulum of eukaryotes, whereas the 70S ribosomes can be seen in prokaryotes as well as chloroplasts and mitochondria. Ribosomes are the site of protein synthesis and is responsible for translation, where amino acids are joined together by enzymes to form a polypeptide chain. Usually, ribosomes are visible as a granular appearance in the cell or as very small black dots. But in this electron micrograph, we can actually see the structure of the ribosome. Now, the first three columns of ribosomes indicates a lateral view of the ribosome. We can see slightly here the cleft where the small subunit and the large subunit meet. On the right-hand side of the image, we can see a frontal view of the ribosome, and here we can slightly see the cleft as well. In this cleft is where the mRNA will pass through to form the polypeptide chain. In the 1960s, Lynn Margulis observed ribosomes inside the mitochondria. These were smaller than cytoplasmic ribosomes. Eukaryotic cytoplasmic ribosomes are ADS, and as mentioned before, bacterial, mitochondrial, and chloroplastic ribosomes are 70S. The similarities between mitochondria, chloroplasts, and bacteria gave rise to the endosymbiont evolutionary theory, and that mitochondria and chloroplast was once free-living bacteria. Other evidence supporting this theory include that mitochondria and chloroplast has their own circular DNA, and they're able to divide independently. If we were to take all the mitochondria or chloroplast out of a cell, the cell would not be able to produce mitochondria or any new chloroplast. The mitochondria, or the so-called powerhouse of the cell, are some of the most abundant organelles in the cell, and up to 2,000 mitochondria can be seen in liver cells. They are usually about 1 micrometer in diameter and bean-shaped, but they can easily change their shape as they move about the cell. The mitochondrion consists of 2 membranes, also called the envelope. The outer membrane contains a protein called porin. These form aqueous channels which allows free movement of water and water-soluble molecules. The inner membrane is folded to form crista, which increases the surface area of the membrane. This membrane acts as a selective barrier and controls exactly what can enter and exit the cell. The space between the 2 membranes is called the intermembrane space and plays a big role in aerobic cellular respiration. The cell is filled with a watery substance called the matrix. Here we will also find ribosomes, circular DNA, and starch granules floating around. The mitochondrion has 2 functions, namely aerobic respiration and lipid synthesis. Cellular respiration consists of a series of reactions that releases energy from energy-rich molecules like carbohydrates and lipids. This energy is transferred to ATP, which is an energy-carrying molecule known as the universal energy currency. The first stage of cellular respiration takes place in the cytoplasm of the cell and occurs anaerobically. This is known as glycolysis. The products of glycolysis then move to the matrix of the mitochondria, where the second stage of the Krebs cycle will occur. In this stage, hydrogens and electrons are produced, which is then moved to the crista of the mitochondria by enzymes found in the matrix. At the crista, across the membrane, a flow of electrons is established by electron carriers. This provides the energy to produce ATP from ADP and phosphate. This is a photomicrograph of a mitochondrion. We can see the outer membrane as well as the inner membrane that's folding throughout the mitochondrion, forming crista. The inside of the mitochondrion has a granular appearance indicating ribosomes. On the outside of the mitochondrion, in the cell cytoplasm, we can also see the rough endoplasmic reticulum, which is easily identifiable by the ribosomes that sits upon it. Chloroplast, in contrast to mitochondria, is much larger at 3 to 10 micrometers. They are found in the green parts of a plant, such as the leaves. This does not mean that plants that have colored or red leaves do not have chloroplast. The red pigment just masks the green pigment from the chloroplast. Similar to mitochondria, chloroplast also consists of a double membrane called the envelope. It also contains its own DNA as well as 70S ribosomes for protein synthesis. Instead of a matrix, the chloroplast is filled with a substance called the stroma. Here, fluid-filled sacs called thylakoids can be found stacked onto one another in structures called grana. The chloroplasts are responsible for producing glucose from photosynthesis. This is done in 2 stages. First, the light-dependent stage. In this stage, light energy is absorbed by the photosynthetic pigment found in the thylakoid. One of these pigments are called chlorophyll. Some of the energy is used to produce ATP from ADP. The rest is used to split water into hydrogen and oxygen. Oxygen will then diffuse out of the cell and hydrogen will move across the thylakoid membrane to produce ATP, similar to respiration in mitochondria. The energy and reducing power produced will then be used to produce glucose from carbon dioxide in the light-independent stage. This stage does not require light and occurs in the stroma. It consists of an enzyme-controlled cycle called the Calvin cycle. The sugars that are produced can then be stored in the stroma as starch grains, alongside lipid droplets that are made from the breakdown of the membrane in the chloroplast. Because light plays such an important role in the functioning of the chloroplast, the thylakoids, as well as the whole chloroplast, is able to change its orientation to maximize light absorption. On the left side of the image, we can see a photomicrograph of a chloroplast. The very dark, almost black stained areas will be the thylakoids that are stacked upon one another to form the granum. On the right hand side of the image, we have a zoomed in photomicrograph of the chloroplast. Here you can see the granum with the stacked thylakoids clearly. You can also see how the lamella connects one granum to another. Enclosing all organelles is a cell membrane. It is extremely thin at 7 nanometers in diameter and has a trilaminar appearance, meaning that two dark lines with a pale interior can be observed. This is because it consists of a phospholipid bilayer. The cell membrane is known as semi-permeable, as it controls substance exchange between the cell and the environment outside of the cell. Sometimes the cell membrane can extend to form finger-like projections called microvilli. These structures are commonly found in epithelial cells and increase the surface area of the cell, which consequently increases the rate of movement of substances across the cell membrane. This is especially beneficial for absorption in the small intestine as well as for reabsorption in the proximal convoluted tubule of the kidney. The cell is able to maintain this structure through the use of a cytoskeleton. The cytoskeleton comprises of 3 different filament types. The smallest of these are the actin filaments at only 6 nanometers. They were first observed in the skeletal muscles and are made up of actin protein. They slide along myosin fibers to enable the cell to contract. This allows the cell to move as well as to obtain a specialized shape, like microvilli. Actin filaments are also responsible for producing the contractile ring in cytokinesis that splits the cell in two. Then there are intermediate filaments that are 10 nanometer strong, rope-like structures that are mainly responsible for strength and support. Lastly, we have the microtubules. These are the largest of the group at 25 nanometers in diameter. Microtubules are made up of alpha tubulin and beta tubulin, which together forms a dimer. These dimers are joined end to end to form a protofilament. When 13 of these protofilaments are arranged in a cylinder, it is known as a microtubule. Microtubules have many functions. They can work alongside intermediate filaments to provide support. They also aid in anchoring membrane-bound organelles into place, but can also move organelles or vesicles alongside the outside surface of the microtubule in an intracellular transport type of system. Microtubules work by being anchored at one end at a site known as a microtubule organizing center, or MTOCs, and then being able to shorten or elongate to perform different tasks. One of these tasks include the aid in the movement of cilia and flagella. MTOCs are located at the base of the cilia or flagella, and here is known as basal bodies. Another well-known example of an MTOC is the centrosome, which is located right outside the nucleus. Inside the centrosome, 2 centrioles can be seen positioned at right angles. These centrioles consist of a ring of 9 triplets of short microtubules. The function of the centrosome is not entirely known, but it is well known that centrioles attach to centromeres on this chromosome to move them about the cell during cell division. This confocal microscope image has different stained areas showing different parts of the cell. The center purple-orange part of the cell is the nucleus. The outside red ring indicates the actin filaments and the green is the intermediate filaments. Here we can see how they stretch across the cell to give its cell shape. Then we have an electron micrograph showing how the centriole is made up of the triplet microtubules. You can see the ring structure with the triplets connected. Then some honorable mentions. First, we have a circular piece of DNA, which usually can be observed in bacterial cells. The smaller piece of DNA is known as a plasmid, and this is the structure that's usually used in genetic modification in biotechnology. Then we have an image of cilia on the outside of a cell. You can see the cilia moving really quickly, helping with movement. It's kind of hair-like structures. Contrasting the cilia, we have an image of microvilli. You can see the microvilli is shorter and thicker. Microvilli in contrast helps with absorption. In this image, there's also a sperm cell notable. And then lastly, we have a cell with a flagella. The flagella is much larger than microvilli and cilia, and it also helps with movement of the cell. In this cell, we can also observe the nucleus in the center of the cell, and surrounding the whole nucleus is a cup-shaped chloroplast. You can see in the chloroplast the different spherical starch granules